Namaskar and welcome to SYBSc student friends. From today onwards, we are going to learn the new topic from the paper number 5, Embryology of the Angiosperm, that is unit number 2nd, Embryo and Endosperm Development, Polyembryony and Apomixis. So, today we are going to learn the structure and development of the monocot embryo. So let us see the introduction regarding this particular embryo and endosperm development. This particular topic or this particular developmental thing is come after the fertilization in the angiosperm, that is union of the male and female gametophyte in the angiosperms. So the development of embryo from fertilized egg is known as embryogenesis. Zygote or oospore is the pioneer stage in the development of the embryo. This particular zygote, which is seen in this particular picture, after syngamy, zygote undergoes a period of rest before entering into the further development. The resting period varies from species to species, that is few hours to few months, and depends on the environmental conditions. First division of zygote is transverse, followed by one or more divisions in parallel plane, resulting into row of cells. This particular thing you can observe in this particular given image. This undifferentiated group of cells is referred as the proembryo, which produces suspensor cell and finally a distinct embryo, which is seen in this particular picture. There is no difference in early stage of development of monocots and dicots, but latter stages show remarkable changes. Hence, these particular monocots and dicots are treated separately. एकदल आणि द्विदल वनस्पती ह्या पर्टिक्युलर रिझन मुळेच सेपरेटली ट्रीट केल्या जातात. Now we will see the today's lecture main part, that is structure of a typical monocot embryo. एकदल सपुष्प वनस्पती ज्या आहेत त्यांच्या एम्ब्रियो हा त्याचं स्ट्रक्चर कसं असतं ते आज आपण बघूया. So this is the entire typical structure of the monocot embryo. Let us see what are the different parts come in this particular monocot embryo. In monocotyledonous, the embryo have only one cotyledon, that is एक दल, ज्याला आपण मराठीत एक दल जे आहे ते म्हणतो. In members of family Poaceae, Poaceae is the grass family, गवताची जी फॅमिली आहे तिला Poaceae म्हणतो आपण. The cotyledons is called scutellum. This is the scutellum region. It is situated towards the lateral side of the embryonal axis. This axis at its lower end has radicle and root cap, which is enclosed in a sheath known as coleorhizae, which is differentiated lower part of the proembryo. The portion of embryonal axis lateral to level of scutellum is the epicotyle. This is the epicotyle region which is shown in the entire circle, which is upper to the coleorhiza. It comprises a shoot apex and few leaf primordia enclosed in a hollow foliar structure known as coleoptile. This is the coleoptile region which is shown in this particular picture of the monocot embryo. On the one side, the coleorhiza gives rise to small outgrowth known as epiblast, which is shown in this particular centrally placed the epiblast. The epiblast represent rudiments of second cotyledon. Now we will see how this monocot embryo get developed. This is the particular picture showing the stages in the development of typical monocot embryo. The example given in this monocot embryo development is Sagittaria. There is no fundamental difference in between development of monocot and dicot embryo, but it is more complex in the monocots. This particular development is complex in the monocot. Embryos are quite different in two groups, that is in monocots and dicots the embryos are quite different. In the present topic, the embryogeny of Sagittaria sagittarifolia belonging to the family Alismataceae. This is the Sagittaria sagittarifolia has been given as one of the example of the monocot embryo development. During the development of monocot embryo, following crucial events were takes place. First one, that is the fertilized egg. This is the diagrammatic picture shows. I will explain this one by one. The fertilized egg, that is zygote. This is the zygote elongates and then divides transversely to form two-celled structure, that is terminal cell and basal cell. This is the second stage showing terminal cell and basal cell. The basal cell, which lies towards the micropylar end, does not divide any further but simply enlarges to form a large vesicular suspensor cell. It may function as a hostorium. This particular cell functions as a hostorium for absorption of the nutrition, while the terminal cell divides transversely and form two cells. Now this three-celled structure is referred as proembryo. This particular three-celled structure is called proembryo. Now this three-celled proembryo have basal cell, middle cell and lower cell. The basal cell functions as a vesicular cell. The middle cell of three-celled proembryo undergoes for transverse division and produces two cells, that is upper cell and lower cell. The upper cell is responsible for formation of hypocotyle and finally root tip, while the lower cell produces shoot apex and 3 to 6 celled suspensor. This particular H stage shows entire the vesicular cell, suspensor, radicle and the cotyledons. Sometimes in few cereals both the plumule and radicle gets covered by sheath developed from scutellum called coleoptile and coleorhiza respectively. The presence of scutellum is the most conspicuous feature of the embryo in Poaceae, that is Gramineae or grass family. The lowermost cell of the proembryo undergoes for a vertical division and produces four-celled structure known as quadrant stage. This is the particular quadrant stage. The octant is formed by transverse division of quadrant, the 8 cells being arranged in 2 tiers of 4 cells. This particular H stage again shows this particular quadrant stage into the 8 cells being arranged into 2 tiers of 4 cells each. This is the octant. This entire octant is seen, which is the 8 cells being arranged into 2 tiers of 4 cells each. The 8 cells then divide by periclinal division to differentiate 8 outer and 8 inner cells. This entire structure shows the 8 outer and 8 inner cells. The outermost layer of octant, 8 outer cells, produce dermatogens, whereas the inner cells, inner 8 cells, produce peribleum and plerome. The 8 cells of the octant, after differentiating the periderm, plerome and dermatogens, divide further to form a single cotyledon. Later, the shoot apex originate from a lateral position situated between the base of the cotyledon and the embryo axis. Now we will discuss this particular development of embryo in the monocots in the summarized form with the help of this particular chart. The zygote developed after fertilization. This zygote, and this particular diagram shows this zygote after the transverse division forms the basal cell and terminal cell, while the terminal cell again transversely divides and form the two cells. These are the two cells. Total 3-celled proembryo is developed. This is the proembryo, and this proembryo forms the basal cell, middle cell, lower cell. The basal cell forms the vesicular cell. This is the vesicular cell, and the middle cell after transverse divisions forms the upper cell and lower cell. This is the upper cell and lower cell. Upper cell forms the hypocotyle and the root tip in the entire plant after the development. Then lower cell forms the shoot apex and suspensor, while the lower cell of the pro-embryo. The second vertical division forms the quadrant stage. This is the quadrant stage of the pro-embryo lower cell. The quadrant stage again transversely divides from the octant stage. The octant stage entirely, this is the octant stage. This is the octant stage, and this octant stage periclinal division occurs in this octant stage and forms the 8 outer cells and 8 inner cells. The 8 outer cells form the dermatogen, while the inner 8 cells forms the periderm pleurum, and from this the one cotyledon gets developed. This is all about the development of the monocot embryo, which develops one cotyledon in the embryo. This is all about the structure and development of the monocot embryo. In next lecture, we will study the structure and the development of dicot embryo. So, thank you.